Morning, students. So in the last classes we discussed regarding this phase 1 reactions and phase 2 reactions. So I explained you about the phase 2 reaction we started, that is first was the glucuronation reaction, second sulfation, and third one is the acetylation reaction. So after three reactions, now we will move on to the next reaction, that is methylation reaction. So this methylation is going to be limited in the body where S-adenosyl methionine, that is the active form of the methionine, which is acting as a methyl group donor molecule. So these reactions are called as transmethylation reactions because transfer of methyl group is going to happen here. So the enzyme which is catalyzing these reactions are methyl transferases. So this how you can see in this slide how the methyl group is going to be transferred with the help of this methyl transferase enzyme. So the compounds which are going to be conjugated by methylations includes nicotinamide. So you can make out here methyl group how it is going to be transferred. So nicotinamide N-methyl nicotinamide you can make out here in this reaction, right? So here paramethyl amino azobenzene, where you can see the transfer of methyl group which is going to happen. So para dimethyl amino azobenzene is going to be the hepatic carcinogen product, the conjugated product which you can see. This is the hepatic carcinogen. So these reactions are mainly catalyzed by methyl transferases enzyme. So the transfer of the main mechanism or the function of this enzyme is transfer of methyl group. So we can see here that is orthomethylation, that was paramethylation. So orthomethylation of estrogen, norepinephrine, epinephrine and their metabolites we can also see. So how these are going to be methylated, and it is going to transfer the methyl group and it is involved in the formation of the particular conjugated product. So coming for the next, conjugation with amino acids. So how these amino acids are going to be conjugated with the molecules, right? So this benzoic acid you can see here, in the benzoic acid is going to be conjugated with a glycine molecule to form epioric acid. So this is the conjugated one, epioric acid. So same way the nicotinamide is going to be conjugated with a glycine molecule to form nicotinuric acid. Nicotinuric acid is going to be the conjugated molecule. So like this, cholic acid and deoxycholic acids are conjugated to form glycocholic acids and glycodeoxycholic acids. So these are going to be the conjugated form of this cholic acid and deoxycholic acids. Coming for the next, that is conjugation with the cysteine amino acids. A few aromatic compounds are involved in the conjugation with the cysteine molecule, that is going to happen in the presence of acetic acid molecule to form mercapturic acids. So bromobenzene is going to be condensed with the cysteine. It is going to be conjugated with the cysteine molecule in the presence of acetic acid. This is resulting in the formation of bromophenyl mercapturic acid. So the bromophenyl mercapturic acid will be the conjugated product which you are going to observe by conjugation with this cysteine amino acid, that is bromobenzene, and in presence of acetic acid. So coming for the next molecule, that is naphthalene. See this how is going to conjugate with the cysteine. Naphthalene is going to conjugate with the cysteine in presence of acetic acid to form naphthyl mercapturic acid. So naphthyl mercapturic acid is going to be the product, conjugated product. If you are seeing the conjugation with the cysteine, some aromatic compound, so the naphthyl is an aromatic compound, it is going to conjugate with the cysteine in presence of acetic acid. So you can see the formation of naphthyl mercapturic acids, respective conjugated product. Coming for the next one, that is conjugation with the glutamine. See how the glutamine amino acid is going to conjugate with the one more molecule, that is phenyl acetic acid. This is also a what is that, aromatic compound, right? Phenyl acetic acid is going to conjugate with the glutamine to form phenyl acetyl glutamine. Phenyl acetyl glutamine is going to be the conjugated form. Understanding? So this reaction is mainly important in the patients of the phenylketonuria. So you can see here in case of phenylketonuria, where you have already studied in your third semester, that is metabolism. So phenyl amino acid is undergoing degradation, that is phenylalanine is getting converted, undergoing degradation to form tyrosine, which is catalyzed by phenylalanine hydroxylase enzyme. So if that enzyme is deficient, where the oxygen molecule is required, there cofactor will be biopterin, right? Tetrahydrobiopterin is going to be formed there. So there, if that particular enzyme is deficient, or the gene which is responsible for the synthesis of that enzyme leads to the formation of, it is going to divert that pathway. Phenyl ketone is going to be formed, phenyl pyruvate is going to be formed, phenyl acetate is going to be formed. If you have remembered so far, I don't know. So these are the three products of that phenylalanine degradation if that enzyme is going to be deficient, that is phenylalanine hydroxylase. So it leads to the phenyl ketone urea, that is that the patient with this reaction which is going to happen in case of the patients with the phenylketonuria, since excess of phenylacetyl, since excess of phenylacetyl, glutamine is going to be excreted in the urine, that imparts a mousy odor to the urine. So you might have remembered that mousy odor which gives the smell for the urine, that mousy odor of the smell because of the presence of this phenylacetyl glutamine which is going to be excreted in the urine because of that enzyme deficient. So in case of those patients, you can come across this kind of reaction which is going to happen, where the conjugation with the glutamine is going to happen and leads to further the excess of secretion of this phenylacetyl glutamine in the urine. Coming for the conjugation with the glutathione, this is the last phase 2 reaction, last reaction of the phase 2. So you can see here in the glutathione, so this glutathione gamma glutamyl cysteine glycine, this is also called as gamma glutamyl cysteine glycine. That means the glutathione consists of 3 amino acids, that is glutamate, cysteine and glycine molecule, which is going to act as a tripeptide. So this is called as a tripeptide, which is consisting of this glutamic acid, cysteine and glycine. So this glutathione is commonly abbreviated as GSH because of the sulfhydryl group of the cysteine, which is a particular part of the molecule, right? So this number of a number of potentially toxic electrophilic xenobiotics such as certain carcinogens and conjugated form of this to the nucleophilic GSH, that is glutathione GSH, which is abbreviated as, in the reactions which can be represented as follows. So this is called as a R is called as here an electrophilic xenobiotic. You can make out in this reaction R plus GSH gives rise to RSG, right? RSG is going to be formed. So you can see here R is considered as electrophilic xenobiotic or the foreign substance. So you can see here the enzyme which is where the conjugation with the glutathione is going to happen. This is mainly catalyzed by the enzyme glutathione S transferases. Main enzyme involved in the conjugation with the glutathiones are glutathione S transferases. So variety of these glutathione S transferases you can come across. These are going to be present in the human tissue, which is going to exhibit or responsible for the different substrate specificities reaction, and which can be separated by electrophoretic and other techniques method, right? So electrophoresis and other technique method, we can separate this particular transferases which is present, other transferases, many variety of this glutathione transferases in case of human tissue. So if the potentially toxic xenobiotics were not conjugated to GSH, so they would be free to combine covalently with the DNA molecule, RNA molecule or any cell protein, and could thus lead to the serious cell damage. So by combining with this molecule, it leads to the further serious damage you can make out here in the cell. So this GSH is therefore which is acting as a very important defense mechanism against certain toxic compounds, which are main toxic compounds such as drugs, it may be, or it may be any carcinogens, right? So this is very important, acting as a very important defense mechanism in order to work against the toxic compounds, either it may be the drugs or the carcinogens. So coming for the effect of xenobiotics, what are the effects of this xenobiotics? Till now we have studied, we have we have seen. So this xenobiotic compounds are the foreign substances, right, which are going to be acting on our body, right? So this mainly going to be detoxified by the liver detoxification process or the detoxification mechanism, which was catalyzed by cytochrome P450 monooxygenase enzyme, right? So you can see the reactive molecules are going to be formed, reactive metabolites. So GSH transferase, that is glutathione transferases, or any epoxide hydrolase which is involved in the conversion of this reactive metabolites to non-toxic metabolites. So this is covalently binding to macromolecule. This reactive intermediates metabolites which are going to be formed are binding to the any covalent binding is going to happen to any macromolecules, or it may be any lipids, proteins or any other molecule. So it leads to the formation of hapten. So you can see here, and cell injury further, and even mutation you can also come across. So further mutation of this leads to the cancerous cells or the cancerous tissues. And apton, in case of this apton which is involved in the uh, uh, which will result in the, you can make out the antibody production. So antibodies are going to be of course produced, which is going to act as a defense mechanism of our body. In the response to the antigen molecule, they are going to produce this antibody molecule in order to uh, act against our body, that is defense, acting as a defense mechanism of our body, which will which you can make out by the cell injury, right? So this xenobiotics, we can see what the cell injury or the cytotoxicity which can be severe enough to result in cell death further. So these macromolecular targets which includes DNA, RNA or any protein, as I said you. So the reactive species of this xenobiotics may bind to the protein, altering its antigenicity property also. And further, I just now said this results in the antibody formation, which can damage the cell by several immunological mechanisms, which grossly but mainly resulting to the normal cellular biochemical processes. By any biochemical processes, you can observe this particular immunological mechanism, which where the antibodies are going to act as a defense mechanism of our body. So this completes the entire topic of this xenobiotics and the endogenous substances. In the next class, we will focus on the cytotoxicity or mutagenicity and carcinogenicity topics, liver toxicity and kidney toxicities. Thank you.